Hey guys, so chapter 4.1, as you see, is the prokaryotic cell. Please raise your hand if you finished chapter 4 homework on hawks. Okay. Okay. So you know, no extension. Alrighty, so we're doing prokaryotic cells. Pro from professional. These are the cells that are different from the eukaryotic cells. So we are not prokaryotes, we are eukaryotes, and we first have to examine the cell at different levels. So when we compare a prokaryotic cell to a eukaryotic cell, what are we going to compare? We're going to compare the structure of the prokaryotic cell and the function of the prokaryotic cell. So cells are the basic building blocks that make up the human body, just like the bricks form a wall. And we know that multiple cells give us tissues, and multiple tissues give us organs, and multiple organs give us organ systems. So the human body is carefully constructed from many specialized cell types, each performing a vital role for growth, development, and for maintenance. So despite their variety, cells from all organisms share certain fundamental characteristics. So you must understand that in the body, you have a neuron, that's the cell of the nervous system. You have a hepatocyte, that's the cell of the liver. You have a cardiac myocyte, that is the cell of the heart. You have a skeletal muscle cell, that is the cell of the skeletal system. You have an osteocyte, that is the cell of the bone. Now, these cells are very different in their structure and function, even though they all have the same, even though they all have the same, even though they all have the same DNA. They all have the same nucleic information. They all have the same genetic information. So the human body is constructed because you have these different systems that have different functions. Therefore, they need to have different cells. But all the cells have one thing in common. What is it? They all have the same DNA. But do they transcribe all the DNA? No. So only certain regions are activated, known as the genes. So certain genes are on, other genes are off. So if a neuron needs a protein, it will transcribe the gene that will give it the protein it needs. If a skeletal muscle cell needs a protein, it must be, it must utilize, if it needs a protein, it must utilize the transcription of a gene that is different from the one that the neuron uses. Is that clear? The DNA has multiple genes. Let's say that's the DNA. So here is gene 1, here is gene 2, here is gene 3, and that's gene 4. So each gene, when you transcribe it, when you transcribe the gene, you get a protein. When you transcribe the gene, you get a protein. So what makes cells different is the set of proteins that the cell has. So even though we all have the same DNA, we don't transcribe. Every cell doesn't transcribe all of it. So for example, there's a big shopping mall. So when all of us walk in the shopping mall, do we go to the same store? So some people go to athletics, some people go to buy some kitchenware, something. It doesn't matter. It just depends on what you need. Same thing. The DNA is like the mall. It's there, but you only transcribe from it the gene that you need. That's why our cells are different. They're not different because they have different DNA. They have the same DNA. They just transcribe different regions that make them who they are. So what makes you who you are is the transcription and the translation of that mRNA transcript. So cells are the smallest unit of a living organism, whether the organism is single cell or multicellular. So multiple cells of one kind interconnect to form tissues, which combine to form organs and organ systems and ultimately an entire organism. So if you look here, you see that you have a pyramid. You have a pyramid. On the base of the pyramid, you will have the cell. Many cells of the same kind will give you a tissue. So many neurons will give you a neuron tissue. Many hepatocytes will give you hepatocyte tissue. So the tissue is made up of the same cells. Now the organ is made up of different tissues. The organ is made up of different tissues. And the organ system is made up of different organs. So for example, we don't have the stomach system or the esophagus system or the liver system or the intestine system. We have the digestive system. So this system is made up of different organs. The esophagus is an organ, the stomach is an organ, the intestine is an organ. So they come together to give me an organ system. And many different organ systems, the digestive system, the nervous system, the musculoskeletal system, all of these systems come together to give me an organism. Excellent. They give me an organism. And many organisms come together to give me many organisms come together to give me a population. Very good. They give me a population. And we said there is an invisible, even more basic level all the way in the bottom. What is that? That is known as the below the cell. That is the subcellular level. Here you have the protons, the electrons, that you have the neutrons, you will have the organelles, you'll have the organelles, and you will have the proteins and the micro and macro molecules. Micro and macro molecules. Is that clear? Are we clear? Okay. So multiple cells of one kind, we said that, interconnect to form tissues, which combine to form organs and organ systems, and ultimately an entire organism. Scientists categorize cells into two broad categories. There are prokaryotic and there are eukaryotic cells. So plants and animals, we are eukaryotic. So bacteria are prokaryotic, but plants and animals are eukaryotic. Now, we talked about the light microscope, and we said that light microscopes use visible lights to magnify specimens, such as the cells. So if you look in that slide, you cannot see anything. But when you look under the microscope, you'll be able to tell the definition. So cells are translucent and staining. So staining is required to distinguish the components of individual cells, but it also kills the cells. So you have to understand that sometimes, we talked about this in lab, that it's not just about the working distance or the total magnification, where you multiply ocular multiplied by objective magnification, also the histological dye or the physiological dye. That now, sometimes you can have the perfect setup, the perfect ocular lens, the perfect objective lens. You can have the perfect working distance. But the problem is you have no proper histological dye, and you will never be able to see the slide, even though you had everything perfect. So don't forget that this histological dye is very important. But one of the negative things is that what does it do? It kills the cell. It kills the cells. Now we're going to talk about magnification versus resolution. So magnification is a process of enlarging an object's appearance. So when you magnify something, you enlarge it and you see in more detail. So compound microscopes have multiple lenses, and the overall magnification equals the product of each lens magnification. So if one lens magnifies 10 times and the eyepiece 10 times, then the total magnification is 100. That's very important to know, that the total magnification must equal the ocular magnification multiplied by the objective magnification. So a higher magnification reduces the resolving power. So the more you magnify, when you zoom in, something that's very far, when you zoom in to see it, you will be able to see, but it's going to be blurry. So they are inverse. The more you magnify, the less resolution you're going to have, which is the ability to distinguish adjacent structures as separate. We talked about that. We said if there's two of you next to each other here, I can see 1, 2. If I go back 10 feet, still 2. 20 feet, 2. 100 feet, I can barely tell there's two students. 200 feet, they're going to look like a one big clump. So now there's no resolution. Resolution is my ability to identify that 2 points are indeed 2. They're not 1. So while immersion oil can improve the resolution at high magnification, so we use this oil in order to help us, because they are enemies. They are enemies. They are like a seesaw. If the magnification is high, the resolution is low. If the resolution is high, the magnification is low. So this oil immersion is going to try to consolidate and help us see things in perspective. So now it's going to improve the resolution. So they're not going to be as enemies. Try to bring them together. So at high magnification, the electron microscopes are ultimately needed for the best resolution to study cellular structure and function in detail. So you have to see that these electron microscopes can see with an excellent magnification and an excellent resolution. This is a light micrograph of the blood taken from a patient with chronic lymphocytic leukemia, and the first image is at magnification of 400 times. The second one is at a magnification of 1,000 times. So here you can see better resolution, but you will see that bad magnification. Here you have an excellent magnification, but really low resolution. Okay, now we're going to talk about the electron microscopes. Electron microscopes utilize an electron beam for higher magnification and resolution. They're going to utilize an electron beam for a higher magnification and resolution. So scanning electron microscopes image surface features, while the transmission electron microscope, there are two types of microscopes. There's the scanning electron microscope, and there's the transmission electron microscope. The scanning electron microscope shows us the surface features, while the transmission shows us the internal structure. That's why they call it transmission, because it goes through. The specimen preparation for the electron microscope is destructive to cells, so enable to have a histological fixation that enables us to see these specimens. We're going to use a histological dye that is going to destroy the cells, but it's going to enable us to see them better, but obviously it's going to destroy the cells. I mean, look at the light microscope and then look at the scanning microscope. I mean, this looks like it's actual image from Google. That's how excellent the resolution is, and that is how excellent the magnification. So the electron microscope is much better in terms of resolution and in terms of magnification, because what does it utilize? What does the electron microscope utilize? Electron beam. Now that's this theory of the cell is you can never have cells except from other cells. Like we are here because our parents are here, our parents are here because our great grandparents are there, our great grandparents are there because their parents were there, our children are here, and then our grandchildren will be there because we come from other cells. We don't come from nowhere. No cells will come except from other cells. You must come from other cells. So Anthony van Leeuwenhoek crafted lenses to observe single-celled, and he called them animalcules. Like, molecule, he called them, interestingly, animalcules. Like these are molecules of animals. But he didn't know the cell at the time. He didn't call them a cell. So he called them animalcules in 1600. Then Robert Hooke, he's the one who coined the term the cell. That's something you need to know. That's something that could be on the exam. I mean, something as big as the cell, who coined the term? What the cell is, that is definitely Robert Hooke. That is definitely Robert Hooke. After viewing a box-like structure, when he said box, he meant that there is like the plasma membrane. Van Leeuwenhoek discovered bacteria and the protozoa, while improvements enabled seeing components inside the cell. So these were the bacteria and the protozoa. Are they prokaryotes or eukaryotes? Prokaryotes. Eukaryotes are animals and plants. Schleiden and Schwann. This Schwann, you will see it again in the nervous system with the Schwann cells, who wrap around myelin, around the axons of the neuron. Proposed the first two tenets of the cell theory. All organisms are made of the cells, and cells are the basic units of life. So the third tenet, so you see all organisms are made of cells, and cells are the basic units of life. And the third tenet, that all cells arise from pre-existing cells. So this is the triad of the cell theory. You need to know the triad of the cell theory. Where are my markers? What is the triad of the cell theory? The triad of the cell theory indicates the triad of the cell theory indicates what's the first tenet of the triad of the cell theory? You need to know that, number one, that all organisms excellent, are made of cells. Then we also believe that the cell is the unit of life. In pathology, you will know that is the unit of death. You will know that the cell is the unit of health, and the cell is the unit of disease. You are healthy because your cells are healthy, and you are sick because your cells are sick. So they are the units of life and death. They are the units of health and disease. And if you are an organism, a plant, an animal, a human being, no matter what your species is, as long as you're an organism, this board here is not an organism. The computer is not an organism. But if you are an organism, you are alive, then you bet your life, 100%, that they are made up of cells. And what was the last tenet that was added? This is the one that I began with. What was the last tenet? Muhammad. Yeah, so all the cells arise from pre-existing cells. All cells arise from pre-existing cells. All cells arise from pre-existing cells. So this is the modern cell theory. Now they expanded from 3 to 7. But all the others are derived from the actual original 3. So let's look at them. All known living things are made up of one or more cells. Number 2, all living things arise from pre-existing cells by division, cell division. We call it cell division cycle. The cell is the fundamental unit of structure and function in all the living organisms. The activity of an organism depends on the total activity of independent cells. The energy flow or metabolism occurs within the cells. That's one of now the definitions of life. You have to be able to carry out metabolism. If you cannot carry out metabolism, you're not considered alive. Now there is a confusion of what defines life. What is life? So they cannot define what life is, but they can define the characteristics that makes you alive. And one of them is you are able to carry out metabolism. You're able to carry out metabolism. The cells contain DNA and chromosomes and RNA in the nucleus and in the cytoplasm. We talked about that the DNA has genes in the nucleus and it has a nucleus in the cytoplasm. It has a DNA in the nucleus and RNA is going to be translated in the ribosomes in the cytoplasm. So now we're going to go over the structure of the cell. What is the cell made up of? All the cells are basically the same in chemical composition in organisms of similar species. Because you know the subcellular level, you got to have your electrolytes, your sodium, your potassium, your calcium. You got to have your enzymes. You got to have your proteins, your lipids for metabolism, your carbohydrates, your nucleic acids. And in fact, we realize that some bacteria have enzymes that we can use to help us survive. Some fungi we can use to help us survive with these antibiotics. You know, back in the day, if you had cut or you had a laceration in your hand and it was infested with like some sort of microbe. So what they do is they used to make a poultice. You know the poultice? They used to make a poultice. They used to make a poultice. This poultice, they used to put in it molded bread. You ever seen bread that went green or black? And then put it right on your injury. And that would heal. So how would that happen? Why would putting molded bread on your laceration, on your bruise, or on your injury is going to lead you to heal? Because the mold has fungus, and the fungi is going to secrete antibiotic, and the antibiotic is going to kill the bacteria. So you use the fungi antibiotic in the molded bread to kill the bacteria infesting your tissue in the inflammation. Okay? And now these poultices are still being used, especially the castor oil poultice, where the women that have breast cancer would make a poultice made up of castor oil and put it under the bra and leave it there for a minute and the cancer is gone. It happened. Some now they use it for prophylaxis. Some women now, to protect themselves from the cancer, from the breast cancer, they make a castor oil and they put it on the bra every once in a while. So it has because it seeps through. It seeps through the skin, and we will see when you do your integumentary system, you will see it's a route of transportation and access to the circulatory system. That's why they give you topical cream, topical antibiotics, because they seep through the skin. So you don't just access the body with your mouth and the oral cavity and intestine absorb into the blood. No, your skin also is a route. Your respiratory system, taking a deep breath, smoking the cigarette. How does that get to the blood? It gets to the blood through the lung. So these are your three routes that will help you access the circulation. Okay. Why did you do that? Okay. So compartmentalization is when you have division. So a school that has all different divisions is highly compartmentalized, while a school that doesn't have HR, doesn't have administration, is just going by. Which you think is better? What do you think is better? The more compartmentalized. So you will see that the bacteria is less compartmentalized. We have nucleus, we have ribosomes, we have the organelles, we have plasma membrane. They don't. You will see what they have. Prokaryotes like bacteria and archaea are single cell, while eukaryotes, that includes animals, plants, fungi, and protists, not parasites, protists. So that is an example of a eukaryote cell. So eukaryotes are not single-celled. We said that eukaryotes are highly compartmentalized and they are not single-celled. But the bacteria are single-celled. All cells have a plasma membrane, a cytoplasm, DNA, and ribosomes in common. So whether you are a prokaryote, whether you are a eukaryote, you gotta have these four. Whether you are a prokaryote, eukaryote, you gotta have these four. What about the virus? What do we consider the virus? Is the virus prokaryote or eukaryote? Raise your hand if you think that the virus is a prokaryote. Raise your hand if you think the virus is a prokaryote. Raise your hand if you think the virus is a eukaryote. Why is everybody not participating? Like everybody, what do you think? What's your intuition? Do you think a virus is a prokaryote? Please raise your hand. 1, 2, 3. If you think a virus is a eukaryote, raise your hand. You think it's a eukaryote. It is not a prokaryote or a eukaryote. It is dead. I want to see somebody. That's how you get extra credit. When you say, no, professor, it's neither. Then I will say, yes.
Said that without saying it.
No, what do you mean you said that without saying it? That doesn't make sense. You can't say I said that without saying it. This is like Yoda from Star Wars. Okay. All right. So the virus is dead, is considered dead, is classified as dead, but it has the reverse transcriptase that it will go back and it will occupy your cell and convert its own mRNA. So the virus only has mRNA. Nobody mentioned virus even, but I'm just so eager to talk about it because that's a big concept. So but remember, any question on the exam would be, which of the following is never shared between the prokaryotes and eukaryotes? So here the law says, if you are a cell, you gotta have these four. Plasma membrane, what's number 2? Cytoplasm, what's number 3? DNA, what's number 4? Ribosomes. Now, if I say, which one of the following is not part of all cells? plasma membrane, the ribosomes, the cytoplasm, the DNA, the nucleus. The nucleus. Or I put the Golgi body. Yeah, yeah. So a prokaryote lacks a nucleus, having its DNA in the central nucleoid region. So it doesn't have a nucleus. The prokaryote has no nucleus. It often has a peptidoglycan cell wall to maintain shape and prevent dehydration. They have a peptidoglycan cell wall. So remember that. These prokaryotes, they don't have a nucleus, and they have DNA in the central, some region known as nucleoid region, nucleoid, but it's not even a nucleus. Don't mix up nucleus with nucleoid. A prokaryote lacks a nucleus, having its DNA in the central nucleoid region. So they often have a peptidoglycan cell wall to maintain the shape and prevent dehydration. So it's very important to know where peptidoglycan cell wall is to maintain the shape and prevent the dehydration. They often have a polysaccharide capsule. That's a slime-like covering made of carbohydrate to attach to the surfaces. So prokaryotes, let's look at their cell size. They are like from 0.1 to 5 micrometer and are much smaller than eukaryotes. That's another distinctive factor. The size, who's bigger than the other? Are prokaryotes more bigger, or are eukaryotes bigger than the prokaryotes? Yeah. So it allows efficient diffusion in prokaryotes, while it allows efficient diffusion in the prokaryotes, while eukaryotes have structural adaptations for intracellular transport. So the cells need small size for an efficient surface area to volume ratio that enables diffusion. So you diffuse better if you have small area. So that concept is very important. You diffuse best if you have a small area. And the larger cells adapt by dividing, increasing the surface area, becoming flat and elongated, or developing organelles. Okay, so they adapt by dividing, increasing the surface area, becoming flat and elongated, squamous shape, columnar shape, cuboidal cell shape. So these are all different cell structures to adapt to a particular function. And some other cells, like the eukaryotes, start to develop more compartmentalized, organelles, like the Golgi body for packaging. Okay, the summary is that there's a unified cell theory. A cell is the smallest unit of life. One or more cells comprise all the organisms, and new cells arise from existing cells, and microscopes are required to study the microscopic cells. There is an electron microscope. There is a that offers high magnification and better resolution than the light microscope. So that's a very definitive concept, that the light microscope is superior in magnification and resolution. So prokaryotes are single-celled, and they have cytoplasm, they have DNA, they have plasma membrane, and they have, what's the fourth thing that prokaryotes have? The ribosome. Okay, 4.1 over. Where's 4.2? 4.2. Now we're going to look at the eukaryotic cells. So eukaryotic cells are very interesting because we have the phrase that form follows function. Means an object's design should serve its purpose. We talked about that in the beginning. Thank you. We said that. Thank you. I'm good. Thank you so much, guys. I appreciate it. So, a knife is sharp because it's able to cut. The hammer, does the hammer need to be sharp? It needs to be dull. So the form dictates the function. The police officer has a lot of weapons, but the cooking chef, does he need a weapon or utensils? Because that's their job. The police officer is dealing with crime, so dealing with dangerous criminals, so they need the weapons. But these ones here, they're cooking. So your job dictates your function and your structure. Your structure. So in architecture, buildings are constructed to facilitate their intended activities. So if you walk into a school, you're going to see a library, you're going to see classrooms. But if you go to a gym, you go to the Planet Fitness, are you going to see a library there? Are you going to see a classroom? Because that is not, it was not designed for that purpose. And you cannot, and people sometimes get to be smart, like they try to do two in one. You know, like I'm gonna make a building. I'm gonna live on the second floor and rent the bottom, and then make some grocery store. It's not gonna work. It's one thing. Are you gonna make it for living, or are you gonna make it commercial? That's you're gonna get the best result. So always compartmentalize based on the purpose. What purpose do you want this building to be for? So the same thing, our cells have different structures because they have different function. So eukaryotic cell structure. So the cell structure of the eukaryote is that it must have, I can't find my markers. This is very important. The cell, this is the eukaryotic cell. That's not working. The cell, not working either. Yes. I'm sorry? Oh, I found them. I found them. Yes. Thank you. I appreciate it. So the cell, the eukaryotic cell has three compartments. What are the three major compartments of my eukaryotic cell? What are the three compartments? Number one, you have to have a plasma membrane. You have to have a fence. You have to have a fence that regulates the permeability of the cell. Then you must have a nucleus that has the DNA, the genetic information. The genetic information, and it must have the genes, the genome, the genes. And in the middle, you have the cytoplasm. Now, the cytoplasm is divided into two compartments itself. The cytoplasm is divided into two compartments. What are they called? The cytoplasm is divided into, I'm sorry? The cytoplasm is divided into the cytosol, and the organelles. So the cytoplasm is like a swimming pool. So the swimming pool has the water and the rubber duckies. right? Floating in the swimming pool. The water and the rubber duckies floating in a swimming pool. Cytoplasm is the same thing. The water in the cytoplasm is called the cytosol. And all of these organelles, the mitochondria, the ribosome, the Golgi body, the endoplasmic reticulum, they are the rubber duckies floating, free floating in the in the cytosol. So all of these organelles, the ribosome, the mitochondria, the ribosome, the mitochondria, and the Golgi body, endoplasmic reticulum, and so on. All of them are free floating in the cytosol. So these are the structure of the eukaryotic cell. 1, 2, 3, and 2 is divided into the swimming pool, into the water and the rubber duckies, the cytosol and the organelles. This is very important. Are we clear with that? Okay. There are numerous membrane-bound organelles, like the endoplasmic reticulum and mitochondria. We're going to go over all of them. These are specialized compartments with specific function, similar to organs in the human body. So examples are the endoplasmic reticulum, the Golgi apparatus, the chloroplast, and the mitochondria. There are several rod-shaped chromosomes. Several rod-shaped chromosomes. So there we go. These are our structures. You have to know the structures of the cell. It's like the exam on the driver license. You need to know 10 out of 10. The stop sign and the yield, you cannot get 9 out of 10. Remember? In order to get the driver's license. This one, you got to know all of them. What is the nucleus? What is it for? So the nucleus has a nuclear envelope, which is a membrane enclosing the nucleus and protein-lined pores that allow the material to move in and out. There's chromatin inside the nucleus where DNA and associated proteins are in the DNA and associated proteins are making up the chromatin. And then you have the nucleolus. The nucleolus is a condensed region where ribosomes are formed. That's a special place inside the nucleus where we make the ribosomes, Then there are the peroxisomes. The peroxisomes, they metabolize the waste. The peroxisomes, they metabolize the waste. So they recycle. So peroxisome makes peroxide, hydrogen peroxide. So if you have something really poisonous and toxic, if you have an agent that is really poisonous, a chemical that is very poisonous and toxic, such as, who can name for me a very toxic and dangerous substance known as the superoxide? So superoxide is a dangerous toxic substance. So the peroxisome has a lot of enzymes are going to mutate and convert it into hydrogen peroxide. So now we made it a little better. Then we're going to convert hydrogen peroxide into water, H2O. We're going to convert hydrogen peroxide into water, H2O plus, into water, H2O plus carbon dioxide. Sorry, plus oxygen. So we breathe oxygen. Why do we need oxygen? We need oxygen for oxidative phosphorylation and for aerobic respiration. Oxidative phosphorylation and for aerobic respiration. So the endoplasmic reticulum, there is a rough endoplasmic reticulum and a smooth endoplasmic reticulum. The rough endoplasmic reticulum is associated with ribosomes. It makes secretory and membrane proteins. There's smooth, then there is smooth ribosome, then there's smooth endoplasmic reticulum. It's important to make the lipids. So you have to look at the structure and be able to tell. Be able to tell who are these particular organelles. There's the microtubules that form the mitotic spindle and they maintain the cell shape. Then there's the centrosome, that is the microtubule organizing center. So these are two cells. You have the human cell and you have the plant cell. The plant cell has this very big bag in the middle. What is it known as? This big bag in the middle here. That is the vacuole. That's the central vacuole. Is filled with cell sap that maintains pressure against the cell wall. Then there is the microtubules. There's a cell wall. Do the eukaryotes have a cell wall? No, they don't have a cell wall. So eukaryotic cells, now we're going to begin with the plasma membrane. So eukaryotic cells, like the prokaryotes, they have a plasma membrane. So a plasma membrane is a phospholipid bilayer with embedded proteins that separates the cell from its environment. So the plasma membrane is a phospholipid bilayer. It's amphipathic. The plasma membrane is amphipathic. What does amphipathic mean? Amphipathic means that it's two in one. It has a polar head and it has a non-polar tail. Remember in philosophy that animal that has a head like a lion but a body like a horse or something like that? You guys been through something like this in philosophy? Some animal that has from the top it looks like I'm sorry? Something like that. Yeah, I don't remember, but something like that. So that's what amphipathic means. It has a phosphate head and it has a lipid tail. Phosphate head, lipid tail. Phosphate head, two lipid tails. Phosphate head, two lipid tails. That's amphipathic.
Does that say polar head and non-polar tail?
Exactly. Or hydrophilic head and hydrophobic tail. And there's actually two of them on each side because you know the cell is surrounded with water. On the outside, the extracellular fluid is aqueous, is water. And on the inside, the cytoplasm is also aqueous. And there is also aqueous and there is water there. Good. So you need to surround water with all the hydrophilic heads. And the tails are non-polar. They are non-polar tails. These are non-polar tails. And the heads, these are polar heads that they can dissolve in the water. They regulate the movement of organic molecules, ions, water, oxygen into and out of the cell. They allow waste products like carbon dioxide and ammonia to exit the cell. So you see that there's movement. Your plasma membrane is very dynamic. The plasma membrane is dynamic. It's constantly changing. There is influx and efflux. If you eat pancake right now, if you take a photo with the electron microscope of your plasma membrane, then you eat cookies or you eat pancake, then you take another photo after the meal, is your plasma membrane going to be looking the same? So what's going to change? It's going to have a lot of pancake on it? No, it's going to have a lot of transporter. It's going to have a lot of glucose transporters. Tunnel. GLUT. GLUT means glucose transporters. They're going to allow the influx and the efflux of the they're going to allow the influx and the efflux of substance. So a phospholipid bilayer with embedded proteins that separate the cell from its environment. They regulate the movement of organic molecules, ions, water, oxygen into and out of the cell. And they allow the waste products like carbon dioxide and ammonia to exit the cell. So you see the heads, the bubbles, the circles, the heads are, guys, this is very important. Look at you have to look at where my mouse is, where I'm pointing. I told you that on the outside, there is aqueous polar substance, right? That's water on the outside. And then there is water on the inside. So all the bubbles, the phosphate heads, the polar heads are touching the water on the outside. And polar heads are touching the water on the inside. In the middle, you're going to have your non-polar substance. So that means if you are at the surface, you are polar. And if you are in the core, embedded here, that makes you, what did I mention about the GLUT, the glucose transporter? The GLUT is going to allow the influx of glucose. So it's like a protein channel. It's like a tunnel, You see the tunnel? The tunnel allowing the influx of molecules. Tunnel allowing the influx of molecules. Does that make sense? So you must understand that the proteins that span, some proteins are on one side. Do you guys see that? These proteins, some proteins are on the top leaflet. You see there is double layer. The plasma membrane has how many layers? 2. Each layer is amphipathic because the head is polar and the tail is non-polar. Now, if you look here, you will see that the heads are polar and the tails are non-polar. So you see that the heads have proteins here. These are known as peripheral because they are on the periphery. They are peripheral. So they can be on the outer periphery or on the inner periphery, on the bottom. Outer periphery or inner periphery. That's the outer periphery. Or the inner periphery. But you see some of them, they are not peripheral. You see where my mouse is pointing? Here. This one is not peripheral. Is spanning both. This is peripheral, is on the lower periphery. And this one is peripheral, on the top periphery. But this one is a transmembrane, like a tunnel. So that one is not peripheral. What do we call that protein? Transmembrane, integral transmembrane protein. Do you see where my mouse is? That circle, what is that? That's a hydrophilic head, and that's a hydrophobic tail. So now, looking at microvilli and celiac disease. So in specialized cells for absorption, like those lining the small intestine, we mentioned that. The small intestine must absorb the milk into your blood, must absorb the food from the intestine into the blood. We absorb the food from the intestine into the blood. The plasma membrane has finger-like projections known as the microvilli to increase the surface area. In celiac disease, an immune response to gluten damages the microvilli, impairing nutrient absorption. That leads to malnutrition, cramping, and diarrhea. That's very important, guys. Are we clear? So what happens in celiac disease? You have an immune disease where there is hypersensitivity to gluten, and your immune system is going to secrete antibodies that are going to destroy the lining of the intestine. So why do we have evaginations like that in the microvilli? Do you see the microvilli here? The microvilli are, you see these evaginations? Why do we have a lot of them? For what? It's not for movement, to increase the surface area.
This is a 5-minute water break to stretch our legs.
Let me finish this lecture. Okay, yeah. Yeah, so I can turn down the recorder and then upload it. Okay, so the celiac disease. Okay. The cytoplasm is the region between the plasma membrane and the nuclear envelope containing organelles suspended in a gel-like cytosol. That is the aqueous portion of the cytoplasm. It consists of 70 to 80% water, but has a semi-solid consistency. It contains glucose, other sugars, amino acids, nucleic acids, fatty acids, and glycerol derivatives. The cytoplasm has dissolved ions like sodium, potassium, and calcium. They host many metabolic reactions, including protein synthesis. Now, this is the nucleus. The nucleus houses the cell's DNA, and it directs the synthesis of the ribosomes and the proteins. So who did we say was responsible for making the ribosomes inside the nucleus? You see this little dark region here? The nucleolus is the region that is responsible for making the ribosomes. Houses the cell's DNA and direct the synthesis of the ribosomes and the proteins. The nuclear envelope is a double membrane structure that constitutes the nucleus' outermost portion. The pores control the movement of the ions, the molecules, and the RNA between the nucleoplasm. There is a semi-solid fluid containing chromatin and nucleolus and the cytoplasm. That's very interesting. Inside the nucleus, you have the chromosomes. These are linear DNA protein structures with specific number per eukaryotic species. So the chromatin is the condensed form during cell division, and it is relieved. So it is tightly coiled and condensed during cell division, but when we are not dividing, because we need to be accessing the genes so we can transcribe. So we can transcribe this DNA, we need to access the genes and perform transcription. So when we are dividing, DNA is tightly condensed. But when we are not dividing, the DNA is spread out and relaxed, decondensed. The nucleolus is an area within the nucleus that assembles and exports ribosomal subunits to the cytoplasm using sections of the DNA that encode for the ribosomal DNA we talked about. Ribosomes are cellular organelles responsible for protein synthesis. This is very high yield. So if you're asked where in the cell do we make proteins, where are proteins made? Ribosome. That's why ribosome is one of the major shared compartments between eukaryote and prokaryote cells. So shared between eukaryote and prokaryote cells are the ribosomes because everybody needs to make proteins. Bacteria cells need to make proteins and eukaryotic cells need to make proteins. So now there are ribosomes that are attached to the endoplasmic reticulum, and there are ribosomes that are free floating in the cell membrane. Cytoplasms around the attached to the cytoplasms, attached to the endoplasmic reticulum. These are known as membrane-bound endoplasmic reticulum cells, and there are also free-floating ribosomes in the cell. Ribosomes are large protein RNA complexes with two subunits. So there is a big subunit and a small subunit. In prokaryotes, they call them 30 subunit and 6 and 50 subunit. They don't add up, 30 subunit and 60 subunit. But for eukaryotes, they are called 40 subunit and 60. For prokaryotes, they are 30 and 50. The small one is 30 subunit, the big one is 50 subunit. While the prokaryotes, the small subunit is 40 subunit, and the larger one is 60. So the mRNA from the nucleus provides the code that ribosomes translate into specific protein sequences. So the cells synthesizing high amounts of proteins like pancreatic enzyme producing cells have high ribosome numbers. You see, the pancreas has a lot of ribosomes because it's going to be making a lot of enzymes. Pancreatic lipase to digest the lipids. Pancreatic amylase to break down the carbohydrates. The mitochondria is the powerhouse of the cell. It produces ATP, and that is the main energy currency through the cellular respiration from chemical energy stored in molecules like glucose. They have double membrane with inner folds known as cristae, and a matrix, and their own ribosomes and the DNA for their specialized energy production roles. Muscle cells are rich in the mitochondria because they want to meet high energy needs, and lack of oxygen is going to lead to lactic acid production instead of ATP. So we have oxygen, we said we have aerobic respiration and oxidative phosphorylation to make ATP. What did we say peroxisomes do? We went over those. Peroxisomes, I'm sorry? Yeah. So peroxisomes are single membrane organelles that carry out oxidation reactions. They carry out oxidation reactions and they do detoxification. So they break down fatty acids, amino acids, and hydrogen peroxide through oxidation reaction. So they detoxify alcohol in the liver, and they have roles in metabolism, defense, and stress response in the plants. Vesicles and vacuoles. Vesicles and vacuoles are membrane-bound sacs for storage and transport. Vacuoles are larger, and vesicles can fuse with other membranes to release their contents while vacuoles remain separate. Plant vacuoles contain enzymes that degrade macromolecules. You see, the mammary tissue is responsible for producing milk for the offspring. So milk is produced inside the cells but must be transported outside the cells, and vesicles are responsible for that. So vacuoles are larger than vesicles, and vesicles fuse with other membranes to release their content, while vacuoles remain static. So vesicles are dynamic, they're moving, while vacuoles are static, they stay in their place. So the plant vacuoles contain enzymes that degrade the macromolecule. So animal cells have centrosomes and lysosomes, while plant cells don't have lysosomes and centrosomes. That sounds like a very important point we need to know. Which cells are never found in a plant cell compared to a human cell? Our animal cell, that is, plant cells don't have lysosomes and centrosomes. Excellent. Plant cells possess a cell wall, chloroplast, and plastids, and they have a large central vacuole. Now, we don't have those. Animal cells don't have vacuoles, don't have chloroplast, and then they don't have plastids. So both animal and plant cells have a plasma membrane, cytoplasm, nucleus, ribosomes. What's happening? Peroxisomes and microtubule organizing centers. So you need to know what is in common between plant and animal cells and what's different. Animal and plant cells have a plasma membrane, cytoplasm, nucleus, ribosomes, mitochondria, peroxisomes, and microtubule organizing centers. So animal cells versus plant cells, these are the centrosomes. The centrosome is a microtubule organizing center found near the nuclei of the animal cell. So they contain centrioles of microtubule triplets. They organize the microtubules and assist in separating the chromosome during the animal cell division. So what's the significance of the centrosome? Yeah, they assist, but they're not essential. So if you don't have them, it's not like you're not gonna divide. So they are supportive. They are accessory to help in the division. Animal cells versus plant cells. So you know the lysosomes. Animal cells have lysosomes. These are for garbage disposal. They have the hydrolytic enzymes, degradative enzymes. They destroy, and like these are like the shredder. These are the shredders using enzymes in acidic environment, and they digest waste, proteins, lipids, nucleic acids. But plant cells, they don't have the lysosomes. Plant cells do not have the lysosomes, and instead they carry out digestive processes in the vacuole. Now, when you compare the animal cell versus the plant cell, you see that the plant cell has a cell wall, while the animal cell does not. Plant cell has a cell wall, the animal cell doesn't. And the main component of the animal cell of the plant cell wall is cellulose. From the name, cellulose is the main compartment for the plant cell wall. So what are plant cell walls made up of? Cellulose. Cell walls provide raw vegetables their crunchy texture. Chloroplasts, they perform photosynthesis in plant cells, producing glucose and oxygen from carbon dioxide, water, and sunlight. Plants are autotrophs that make their own food. Plants are autotrophs that make their own food. So autotroph, auto is you originate, you make yourself, you execute on your own. Auto. Troph mean food making. So they make the food relying on them by themselves. They use sunlight to make the food. So the chloroplasts perform photosynthesis. That's important. Who's responsible for photosynthesis? It is the chloroplast. They produce oxygen and glucose from carbon dioxide and water and the sunlight. And plants are autotrophs that make their own food. Chloroplasts contain an inner membrane enclosing a set of interconnected flattened discs known as the thylakoids, where chlorophyll is located to capture the light energy for photosynthesis, with the surrounding fluid known as the stroma. With the surrounding fluid known as the stroma. So the endosymbiotic theory is the origin of the mitochondria and the chloroplast. So mitochondria and chloroplast originated from an aerobic that uses oxygen for respiration and autotrophic, makes its own food, bacteria. So you had bacteria that makes its own food and you have autotrophs and you have aerobic organelles that combine together, being engulfed by host cells in an endosymbiotic relationship. So endosymbiosis means it involves one organism living inside the other in a mutually beneficial way, with ingested bacteria evolving into specialized organelles over time. Supporting evidence includes similar sizes and the presence of DNA and ribosomes in the bacteria. So mitochondria and chloroplast, as well as the endosymbiotic relationship seen in the nature today. Lastly, the central vacuole. So the plant cells have a central vacuole occupying most of the cell's interior area that regulates water concentration. So the water loss from a vacuole causes the wilting due to the loss of the supportive cell wall. The central vacuole allows the cell expansion by accommodating more water as the cell grows larger without requiring the synthesis of additional cytoplasm. So this is a summary that eukaryotic cells are larger than prokaryotes. They have true nucleus, they have membrane-bound organelles, and they have linear chromosomes. The key organelles include the nucleolus, the ribosome assembly, ribosomes for protein synthesis, mitochondria for ATP production, and peroxisomes for oxidation reaction. Vesicles and vacuoles function in storage and transport, with plant vacuoles also degrading the macromolecules. Animal cells have centrosomes with centrioles involved in cell division and lysosomes as digestive organelles. Plant cells have cellulosic cell walls, chloroplasts for photosynthesis, and a large central vacuole. Okay, please take a 15-minute break and come back at 11:15. Yes, you can.
But then she's like, well, let me look at it now, and let you know if you have to change it. No, people. Yeah. No, even when like the college, she don't know who has to pay something. But like, I feel like she's being here long enough to know that. She didn't know, no, not. No, I'm not like knowledge. She just pay my dollar. No, she's been here. But like some of the stuff she's not too sure about. It's like when you she don't know about anything. Yeah. Together. That was what I was trying to say. That I don't know. And she thinks that she was trying to be rude. Yeah, I was trying to say the same thing. And then 5:30. So I'm in 1:30. Yeah, I was trying to let her know that. Then we don't need exactly. Yeah, just like a lot of people even find it difficult to kind of upload how to. She's not doing all of that. I tried to say, getting the support from my colleague was like, no, I was trying to like the support. Nobody know why I do have to teacher, right? You expect. I had like 5 people complaining about the same thing. So I expect if I come out and tell them, I should have some kind of support. It will make sense. Yeah, but it's like you're the only one saying it, and everybody's like, oh no, let's go to the next. How can we go to the next step if people don't get the first step? So you realize the first hour of my class is just people asking questions. No, that's I'm not the kind of, I was like, fuck it, I'm gonna do whatever I need. And she always go like, we know when we left there was one person, I don't know who she was. She wrote in the group chat that, oh, it's an accelerated program, so she's gonna be moving fast. It's like that. I find it. I know it's an accelerated program.
the farm, like, with the correct information. So,
Your hours is like an hour.
Hours too. Hours too.
No.
And people ask the same question, but over.
And I don't make those.
Okay. No, yeah, because we don't know where it is.
They don't know, but they. Right. You ask me, they want to answer. Yeah. Let me see. I feel like.
You're done.
And then the next day, put the little screen, not just kind of something, but what
Already know.
Like ATI, show you how to
One change, you guys like, we all have to be on the same page. Then we all can complain to her, that, okay, this is what I'm struggling with. So maybe let's bring it this way in my phone. Like if I was the class, somebody in the class was like, oh, it's like, oh yeah, give me some people find it difficult to catch up, some are fast. We all understand, so if it's easy for you, it doesn't mean it's easy for me. So you know,
I'm a class.
both of us. So it's better going.
Ghetto.
Y'all trying to learn. I should know what you're gonna
I'm glad they start arguing, but now they're like, I'm taking care. When they start arguing, that bitch was a she really teach her. So I can read. She just read like PowerPoint and explain it. You know,
But she's saying things like,
yeah.
They're all different.
and it's like, feedback.
But it's not. I didn't have to learn. I'm here to learn. I'm here to. Right. She didn't give me all the I know it's no, yeah. And everything. And then I agree with them. That's how it looks. And I can have one generous one. I've never bring up. And like I said, don't want it, guys.
What class is that? Wait, who what? For,
I hope that's not the case, but she didn't bring that up though.
Yeah, I think she started there.
Yeah, but that's the case, like.
Yeah.
That's crazy. She probably don't know, cause she really not good.
Yeah.
Nah, but she didn't mention it though. I don't know if that was her. She don't mention it yet, so let's do it. For the Iron team, whatever. Just questions. Yeah, that's what yeah. We're not learning I think she did was something. I remember we really did last night. And not saying she didn't based on it.
We doing 4.3. We did 4.2, right?
I should not do that. I'm trying to
Yeah. I have like you because some kids posted like they took a photo of the computer to the fucking She's now here. You can't
They're the same. I thought that. Oh, you no, like I yeah, right. And then you come up with
what? You said, inshallah, you're gonna open it for me when you think.
I'm gonna open. No, now do the
box?
midterm first. That way the virtual, right? No, leave that till after the midterm.
The McGrid? McGrid? Okay.
Yeah.
The one I'm in. Okay. Yourself on that one. You probably won't. You can look. You can look. You won't get it. Because they make you trying to say whatever the background. Yeah. So they feel like she got jokes. So I know she got what Christian wants to be on. Yeah. So that guy you're saying, oh yeah, this guy like the kids for her. Yeah. So the only thing and I think that's like 25. So I think somebody else. One or two, 2 and 10. Nobody. That's it. Yeah, that's okay. I don't want it. Yeah. I don't think so. I'm telling you, she don't cost nothing like that.
Okay, all right. So we're gonna do the 4.3 now. Okay, so now we're gonna start the endomembrane system. So we're gonna now list the components of the endomembrane system. And the endomembrane system is a group of membranes and organelles in the eukaryotic cells that work together to modify, package, and transport lipids and proteins. So the endomembrane system is like the Amazon, just shipping and packaging proteins. I remember back when I was in college, when I was a freshman, we had a question, which of the following is not part of the endomembrane system? And obviously I got it wrong. So not until I read this chapter now, 20 years later, then I realized the endomembrane, because it's not something big in medicine. It's big in biology. But it just stuck to my head because I never came across it again. Beside that course, it was biology lab also. It was biology 101. So one of the questions was the endo. So now look, who is the endomembrane system? The nuclear envelope, the lysosomes, the vesicles, the endoplasmic reticulum, the plasma membrane, and the Golgi apparatus. So everybody here who participates in making this vesicle, in making these proteins, meaning from the nuclear envelope, the lysosomes, because they cut and they align and shape properly these vesicles, and there's a endoplasmic reticulum, and there's a plasma membrane, and the Golgi apparatus. Okay, the endoplasmic reticulum is a network of membranous sacs and tubules. The endoplasmic reticulum is a network of membranous sacs and tubules that is continuous with the nuclear envelope membrane. It is rough. So the rough endoplasmic reticulum modifies the proteins. There is a smooth endoplasmic reticulum. It synthesizes the lipids. So these are part of the endomembrane system. Your endomembrane system is all about your ribosomes, the vesicles, the lysosomes, and the Golgi body, and the plasma membrane, and the nuclear envelope. Everything that has a rounded vesicular shape that allows for transport, exocytosis, and endocytosis. It modifies proteins by folding and adding side chains, which are secreted or incorporated into cellular membranes, and it makes phospholipids. The smooth endoplasmic reticulum lacks ribosomes on its surface, and it's continuous with the rough endoplasmic reticulum. It synthesizes carbohydrates, lipids, steroid hormones, and detoxifies medication and poisons, and stores calcium for muscle contraction in muscle cells. So these are the roles of the endomembrane system organelles. These are the smooth endoplasmic reticulum lacking ribosomes on its surface, and is continuous with the rough endoplasmic reticulum. It synthesizes the carbohydrates, lipids, steroid hormones. It detoxifies medication and poison and stores calcium for muscle contraction in the muscle cells. The Golgi apparatus. The Golgi apparatus is a series of flattened membranes that sort, tag, and package the lipids and the proteins for distribution. So they modify them for sorting by adding a sugar chain. So the Golgi apparatus is comprised of a series of flattened membranes that sort, tag, and package lipids and proteins for distribution. It modifies them for uh, sorting by adding a sugar chain. Then it tags them with a phosphate group or other molecules to travel on the correct destination. The secretory vesicles bud from the transphase of the Golgi body. Lysosomes use hydrolytic enzymes to digest macromolecules, recycle organelles, and destroy the pathogens. To digest macromolecules, they digest macromolecules, recycle the organelles, and destroy the pathogens. So these are the police officers of the cell. So because the lysosomes, they are the police, they have acid inside them. They have proteolytic enzymes. They have degradative enzymes. They have lysosomal enzymes that once they engulf and they swallow, once they swallow any particular microbe, they hydrolyze it, they digest it, and they destroy it. Now, when you look at the macrophages are white blood cells that engulf the pathogens through phagocytosis and break them down in the lysosome. You see, this is the bacteria. You see where my mouse pointing? That's the red bacteria that must be destroyed. And this is the neutrophil. So the neutrophil is going to swallow and gallop the bacteria, and inside there is a phagosome. There's a formation of a phagosome, and inside you will have the lysosome is gonna fuse with this phagosome. What's a phagosome? A phagosome is a vesicle that phagocytoses the bacteria that must be destroyed. Once it's phagocytosed, then you will see that you will see particularly these lysosomal vesicles are gonna gallop the phagosome. So now you're gonna have a phagolysosome. They're gonna gallop up and swallow this bacteria, destroy it. After they destroy it, it's going to come out. So one of the videos that I'm going to show you now on my channel, I talk about that really well. So if you go, I'm going to assign it to you after the midterm, because now I want you to focus mainly on preparing your homework, your study guides. But if you see here, this is an example of the there we go. Right here. If you see this, actually not that much, like 70 pounds. I lost 70 pounds. After the surgery, that's not a lot. It should be 100 after the surgery, because I did the sleeve, yeah.
Yeah.
So not good, not good, very bad. So look at this. So what do I have here? Do you see? What is this? This big blue. Do you see that?
Yeah.
So the big blue, that's the, you see the microbe? You see where my mouse pointing? That's the microbe. The microbe actually binds on the outside. That's step one. The microbe binds on the outside. Then after it binds on the outside, it is internalized in the vesicle. Then it is now you have this phagosome. The phagosome is the vesicle that has the microbe inside it. Then who's this that has the knife? You see the knives? Who has these knives? The lysosome. Now they're gonna come together, the lysosome and the phagosome, to give you a lysophagosome or a phagolysosome. Then they're gonna go in and they're gonna destroy the microbes, and then they're gonna send them out with the reactive oxidative species. Do you get it? So this is a video you're gonna have to watch and summarize.
When is this summarized?
After the midterm, I'm gonna send it out.
The activation.
You guys are shocked?
Yeah.
Nah, you look mad.
This was like 3 years ago.
Nah, you look mad.
It's just the weight.
Your face is even that fat.
Well, yeah, because obesity. This is called truncal.
The
And-
second button on your shirt, bro.
Yeah. Yeah, I know. It's not the second. Yeah, it is. Yeah, my mom told me that, but I used to think I'm the muscular. I used to think I am 50 Cent. It's not good. But that was not muscle. That was fat.
How much percent body fat was you right here?
Do you think I counted? I liked it. So but it's not good. You got to take care of your health, whatever it takes. You got to take care of your health really well. But the knowledge is the same. The knowledge, yeah, I know. My eyes were like, but I didn't change that much.
The whole skin tone doesn't change.
Okay, another thing about the skin tone. I was in Sudan, my country, the equator. It is so hot. I was baked there. Whenever I come back for vacation, I am so tan.
You from Sudan?
Yeah.
Oh, really?
Yeah. You're from Guinea. Ghana. Oh wow. You're from Guinea. I know Fatima's from Guinea. I know Fatima's from Guinea, and Ilham's from Ghana, and Mahadur is from Pakistan.
I'm from Guinea too.
And Tyra too is from Guinea.
You from
Yeah. You don't know what?
Yeah.
Yeah, she do. Okay, alrighty. So you're gonna watch that. You're gonna watch that and make a summary so you get full credit. It's gonna help you understand how the phagosome is gonna gallop up and swallow and degrade.
So watch this and then do what you grab from the back.
Yeah, not now. After the midterm. Now I need you busy with the midterm. That's it. After the midterm, then we're gonna do that. Okay?
Just study what you put, that test you put in the.
Yeah. Study the test and the PowerPoint slides, and you're gonna do the homework. Yeah, and then I'm gonna give you now part 2. Remember that was part 1. Now part 2 coming out. So I'm happy that you guys did well on part exam 1B. Now it's no more AB. It's gonna be the same.
Question.
Yeah.
Is the 1A gonna just disappear?
No, it's gonna stay there, but it's gonna average. So the 90
How
and
does it average?
No.
1B is nothing.
no. No. Data can never be deleted. What?
Wait, how are you gonna average it when you look at it on campus? The second grade doesn't seem like it's gonna be that.
No, I'm gonna fix it up. I'm gonna line it up with Canvas. I'm gonna call them. We have a conference. We're gonna align. Don't worry. Worry now about learning and doing your best, and the grades are gonna come. Yeah, that's the course. Yeah. Okay, guys. Now we're continuing with the endomembrane system. So the endomembrane system, remember, what does it have? It includes the nuclear envelope, the lysosomes, the vesicles, the endoplasmic reticulum, the Golgi apparatus, and the plasma membrane. So these are the ones that are the endomembrane system is responsible for the packaging and the sorting and preparing and packaging all of the proteins and the lipids that must be exported, imported, and shuffled and moved around the cell. So the rough endoplasmic reticulum modifies proteins and synthesizes phospholipids, while the small endoplasmic reticulum synthesizes carbohydrates, lipids, steroids, and detoxifies the substances. The Golgi apparatus sorts, tags, packages, and distributes the lipids and the proteins. Lysosomes digest macromolecules, they recycle the organelles and destroy the pathogens. Okay, now the last part of chapter 4. Okay, so now we're going to talk about the cytoskeleton. The cytoskeleton, from the word cyto, what does cyto mean? What does cyto mean? Cyto means cell. Remember cytology? Very good. Cytology is the ology is the knowledge of, the learning of, the science of the cell. So cytoskeleton, that is the skeleton that gives the cell its frame and its structure, is within the cytoplasm and consists of microfilaments, intermediate filaments, and microtubules. Microfilaments, intermediate filaments, and microtubules. It maintains cell shape, it anchors the organelles, it allows the movement of cytoplasm and vesicles, and enables cell motility. So the cytoskeleton is within the cytoplasm and consists of microfilaments, intermediate filaments, and microtubules.
So is it like the skeleton of the cell?
Exactly, that's what it's called, the skeleton of the cell. But it's not made of bone. It is made up of microfilaments, intermediate filaments, and microtubules. It maintains the cell shape, it anchors the organelles, allows the movement of the cytoplasm and the vesicles, and enables cell motility. These are the microfilaments. You know, the cell need to move, cell crawling, cell walking. So the cell has appendages that enable the cell to walk, enable the cell to move from one place to another. So microfilaments, these are the narrowest fibers of the cytoskeleton, and they function in facilitating cellular movement and motility. They have a diameter of approximately 7 nanometers, and they are comprised of two intertwined strands of the globular proteins, actin, and they also known as actin filaments. So remember, microfilaments, they are the narrowest fibers of the cytoskeleton, and they function in facilitating cellular movement, crawling. They have a diameter of 7 nanometers. They are comprised of two intertwined strands of the globular protein actin, also known as actin filament. Also known as actin filament. So ATP powers actin to assemble its filamentous form, which serves as a track for the movement of a motor protein called myosin. So it's very important for you to realize that this ATP molecule is central in powering the actin to assemble. ATP powers actin to assemble its filamentous form, which serves as a track for the movement of the motor protein called myosin. It allows the movement necessary for cell division, cytoplasmic streaming, and muscle contraction. Microfilaments also provide rigidity and shape for the cell. They enable the cells to change the shape and movement by their rapid assembly or disassembly. White blood cells depend on this to find phagocytize pathogens. We talked about that. Cells crawl. These immune cells, they have to walk over and crawl to the microbe in order to engulf it, bring it in, and destroy it. Intermediate filaments. Intermediate filaments are composed of wound fibrous protein strands with a diameter of 8 to 10 nanometers. Their diameter is between that of microfilaments and microtubules, hence their name. They comprise various types of fibrous proteins like keratin for hair, nails, and the skin. Intermediate filaments serve a purely structural role. So they bear tension to maintain the cell's shape and form. So these are the intermediate filaments, and they are important. So you need to note that the cytoskeleton of the cell is made up of different types of cytoskeletal proteins. Some of them are myosin, some of them are actin, some of them are microtubules, some of them are microfilaments, others are intermediate filaments. They are composed of wound fibrous protein strands within a diameter of 8 to 10 nanometers. Their diameter is between that of microfilaments and microtubules, hence their name. They comprise various types of fibrous proteins like keratin for hair, nails, and the skin. Intermediate filaments serve a purely structural role. They bear tension to maintain the cell shape and form. They anchor the organelles like the nucleus in place within the cell. Microtubules are small, hollow tubes that help the cell resist compression. They provide a track along which the vesicles move through the cell. They pull the replicated chromosomes to the opposite ends of a dividing cell. So they comprise the structural elements of the centrioles, the flagella, and the cilia. Microtubules are comprised of tubulin dimers, and they have a diameter of 25 nanometers. These are the microtubules. These are a special type of cytoskeletal proteins. So you have to understand that cilia and flagella have microtubules in them. Flagella are found in the bacteria, but humans also have flagella in the form of a sperm. So if a man, they found flagella in the man, that is either because of the sperm or because they were infected with bacteria. If a woman, they found flagella in the body of the woman, that's definitely because maybe there was a sperm in sexual intercourse, or there was infection with bacteria. We don't have flagella as part of our body, but we do have cilia. Cilia, that is in our respiratory tract. They are like the mop, the bathroom mop. They mop down mucus. So they do a power stroke and recovery stroke. Power stroke, recovery stroke. Powering and moving the mucus down. That's why diseases with a mutation in the formation of the cilia, they're going to be unable to move the mucus, and they're going to have a lot of mucus build up on their lung. They're going to be coughing all the time. So you have to be careful. So cilia are a short hair-like structures that extend from the plasma membrane and move entire cells or substances along the cell's outer surface. They are found in the fallopian tubes and the respiratory tract. The flagella and the cilia have a common 9+2 microtubule arrangement. This consists of 9 microtubule doublets surrounding the 2 central microtubules. This is very important to realize, guys. They have, if you see this structure here, 9 plus 2, they have 9 doublets, 1, 2, 3, 4, 5, 6, 7, 8, 9, plus 2 in the middle. So that's the structure of the flagella and the cilia. So the flagella are the whip-like structures that enable the sperm to move. Okay? While the cilia, they are hair-like structures. They are not whip-like. They extend from the plasma membrane and move the entire cells or substances along the cell's outer surface. So they are found in the fallopian tubes and the respiratory tract. Flagella and cilia have a common 9+2 microtubule arrangement, and they consist of 9 microtubule doublets surrounding 2 central microtubules. Components of prokaryotic and eukaryotic cells. Plasma membrane, obviously we said it separates the cell from the external environment. It is found in all of them. Remember we said that all cells must have plasma membrane. All cells must have DNA. All cells must have ribosomes, and all cells must have cytoplasm. Now, you see you see not all cells have nucleoles. Nucleoles are like the boro, where you have inside the nucleoplasm. The nucleoplasm is a combination of the DNA, the chromatin, and associated proteins, and some other proteins there to help, like the polymerases that polymerize the DNA. So you have to go over this figure and decipher which cell component is found, whether in prokaryotes, whether in animals, and whether in plant cells. So the cytoskeleton has 3 protein elements: the microfilaments, the intermediate filaments, and the microtubules. The microfilament, the intermediate filaments, and the microtubules. Microfilament, the intermediate filament, and the microtubules. So the microfilaments provide rigidity, shape, and they enable the movement with myosin. Intermediate filaments bear tension and anchor the organelles. Microtubules provide resistance to compression, as well as they transport vesicles and they separate the chromosomes. Microtubules form centrioles, the flagella, and the cilia. Okay, so that was that. That was 4. So now we're gonna wrap up